Hello, and welcome to a bonus background lecture. This is going to cover some chemistry, biochemistry. This is a material that I would teach in a general biology class. And I know for most of us, it's been some time since we had general biology. And so I thought I would just give a refresher for those who would like it. This is going to go along with the gastrointestinal tract information. And the reason I'm going into some detail is in order to understand the process of digestion, that is breaking down molecules, and absorption, and how certain more broken down molecules can be brought into the gut, and how some molecules, if they're not broken down correctly, they cannot be absorbed. All right, so the four major organic molecules, organic in the chemistry sense, meaning contains carbon. These are often referred to as the macromolecules or the macros. Three of these four are what people keep track of in their diets. So carbohydrates, proteins, nucleic acids. I put these in parentheses because they are a macronutrient, but nobody ever talks about, I'm not sure if I'm getting enough nucleic acid in my diet. Yeah, if you're eating anything that was ever alive, then yeah, you're getting plenty of nucleic acids. You have enough. And then the fourth of the macromolecules, but the third of the things that people keep track of are the lipids. In common parlance, people call these fats, but that's a subcategory of the lipids. All right, and so we're going to go over them one at a time. We'll do carbohydrates first. The function is for short-term energy storage. The simple carbs are also known as sugars, but sugars belong to the larger category known as carbohydrates. Sugars are made of one or two subunits. If it's one unit, it's called a monosaccharide, meaning one sugar, or disaccharide, two sugars. I'll give you an example shortly, but you'll see it's sucrose, which is the most common of the disaccharides. It's a glucose and a fructose, two monosaccharides hooked together. And then what we call complex carbohydrates, also known as polysaccharides, multiple units. They're made of hundreds of units. Specifically, they're made of hundreds of glucose units. And this is why when we talk about diabetic patients, and sometimes they have trouble understanding, that even though it doesn't taste sweet in their mouths, complex carbohydrates break down to sugar. They just break down to glucose, and it takes almost no time for that to happen. Excuse me. Sorry. Plants use a couple of forms of complex carbohydrates. The two most common are starch, which is what we're most accustomed to in potatoes and wheat. The other one is cellulose, and these are used for structure in plants. This is what we refer to as fiber when we talk about our diets. And the difference between them, they're both made of these monosaccharides called glucose, and they're hooked together a certain way. We humans lack the enzyme necessary for taking the glucoses apart from cellulose, so we can't digest it, which is why it goes through us pretty much intact. Yeah, I'll use this example. If you've ever eaten corn, and then the next day you look in the toilet and say, that still looks like corn. Yeah, that's the cellulose that you weren't able to digest. The starch you got out of there just fine, and you digested that, but the cellulose stays pretty much intact. This also happens with other vegetables. Yeah, corn's the easy one to talk about. So we eat a lot of starch. We can eat a lot of starch, but we don't store it as starch. We break it up into the individual glucoses, and then we put it back together in a different form. We call it glycogen, and we can store that mostly in our liver, but we can also store some in our muscles. And I'll show you what those look like. So let's see. Here is table sugar. It is formed from two monosaccharides, a glucose and a fructose, that get put together into this sucrose, which is a disaccharide. We all have the enzyme called sucrase. It breaks up the glucose from the fructose. That's digesting the sucrose. Yeah, I'll mention it. So there's another disaccharide that some of us can digest and some of us can't. And that's lactose. Yeah, lactose. It's a glucose and a galactose. It's two simple sugars. Some of us have the enzyme lactase to break those apart, and people who don't have the enzyme lactase, they can't break lactose apart. They are so-called lactose intolerant, and we'll talk about that during the GI lecture. This is examples of polysaccharides. These are the two we talked about that are present in plants and in our food. Starch. This is the glucoses. They're all lined up. They do a little bit of branching, but they're all lined up together. And we all have the enzymes that can break these glucoses apart from the larger starch molecule. It's the cellulose. This is harder to see, but the glucoses are, one's right side up, the other one's upside down, the next one's right side up, and so on. That's not super important, except that we don't have the enzyme that can break these apart from each other. And so this is why cellulose passes through us without being broken up. And then in the middle, we have this branching form. So we can consume starch, we pull the glucoses apart, and then if we have to store it for any amount of time, we put it together in this more branching pattern. You can get more glucoses into a smaller area, a smaller volume, putting it together like this. And that's what we do, we animals. That was the first of the macromolecules. The second one we're going to deal with is proteins. And we've talked a little bit about these. They're largely for structure. They have jobs. These are things like actin and myosin, tropomyosin, troponin. They have jobs. Proteins that can help make reactions go, that's what we call enzymes. The units that proteins are made out of, just like carbohydrates are made out of saccharides, are made out of these individual sugars, the proteins are made out of individual units called amino acids. Each of the amino acids has 3 parts to it, and this is where the name amino acid comes from. It has an amino group, that's the part with the nitrogen. A carboxyl group, it's also known as an acidic group, that's why it's called amino acid. And then the so-called R group. There's 20 different R groups. That's the difference between the different amino acids. When you put a bunch of amino acids together in a strand, that thing's called a polypeptide. Sometimes you'll hear it called a peptide chain. It just means a strip of amino acids. Why it's called that is the bonds between the amino acids are called peptide bonds. I'm not going to go into a lot of detail this. I'll remind you in case you remember different levels of structure. There's 4 levels of structure. Primary structure is the order in which the amino acids occur. And we've talked about this a little bit. If there's a mutation in the gene that codes for this protein, it's going to mess up this primary, the order of the amino acids, and that has consequences for how the thing is shaped at the end. So secondary, tertiary, and then finally quaternary, if you have multiple polypeptides that come together to form a functional protein. For those who want to see it, here's an individual amino acid. It's got an amino group on one side, a carboxyl group on the other, and then this is the part that varies, this residue, this R group. It can be big, it can be small. There's 20 different ones. This is what we mean by primary structure. You've got an amino end, and then all the different beads on this chain, if you will. There's 20 different ones to choose from, and it's the order of those 20 different kinds of beads. Primary structure then gets twisted some more and then folded some more until finally Yeah. So going back to this. So when the thing is folded up, how does it hold this shape? It all has to do with these different amino acids in a row. Some of these amino acids will bind with each other across the way, and that's what holds the position of this protein. And what amino acids will bind to each other can change depending on how acidic the environment is or how basic it is. And that's why that maintaining the 7.35 to 7.45 pH in the blood is so important, because when you mess around with that, you mess around with the overall shape of all these different proteins, and that impacts their ability to do their jobs. And then finally, okay, quaternary level structure. You've seen one of these diagrams before, because I've shown you when we were talking about hemoglobin. This was the diagram that I took. But all the proteins are like this. Collagen, it's got a particular shape to it, and it all comes down to what's the order of the amino acids and which ones hold on to each other and which ones don't. Okay, now we're on to the third of the macromolecules. This one I'll talk about here. We're not going to talk about it much in the GI tract lecture, but nucleic acids, their function primarily, we've talked about them as being part of the genetic information. This is the DNA and the RNA. Also, we've alluded to it, the part of the energy currency, that is ATP, is actually a nucleic acid. It's a nucleotide. All right, so the genetic information is carried in the DNA and the RNA. That's what the NA stands for, nucleic acid. So deoxyribonucleic acid and ribonucleic acid. The individual units here are nucleotides. Just like amino acids had 3 subparts, so do the nucleotides. They have 3 subparts. There's a sugar, a phosphate group, and a nitrogenous base. In this case, there's only 4 different, there's a total of 5 different nitrogenous bases, but within DNA, there's only 4, and then within RNA, there's only 4. And then the energy currency molecule, you know it and love it, it's ATP, adenosine triphosphate. Here's a close-up of a nucleotide. We've got a sugar. Pentose just means it has 5 sides to it. Sugar phosphate group, here's the phosphate group, and the nitrogenous base. And you put a bunch of these nucleotides in a row, and you get a polynucleotide. All right, without going into a huge amount of detail, if you look at the nitrogenous base, that's the kind of golden colored thing over here on the right. Notice that some of them have a single ring and some of them have 2 rings. There are some differences. There are some, let me show you. You know the A's, the T's, the G's, and the C's of DNA. This is where adenine, A's and T's, thymine, these bind to each other. And G's and C's go together, guanine, cytosine. You may remember this. Why I'm even mentioning it is because at some point we're going to talk about gout, and we'll talk about purines and diets high in purine. These are the purines. So we'll get back to that. What else? Again, those who love detail, the difference between deoxyribose and ribose, so DNA and RNA, is just this. In the sugar, the ribose sugar, believe it or not, it comes down to this. Do you see this a hydroxyl group here, the OH? Here, instead of an OH, it's hydrogen by itself. That's it, deoxyribose. It's missing this one oxygen, and this is just regular ribose. And that makes an enormous difference in the structure of the thing. I'm never going to test you on that. Here's DNA, making new strands. You use the nucleotides like a little unit. You just click on the correct one. And I think that's it on nucleotides. And that brings us to the final category, and these are lipids. And lipids are a big group and they're pretty varied. So various different functions. What they all have in common and why they're put in the same bucket is that they don't dissolve in water. They're all hydrophobic. The three main types of lipids are triglycerides. This is, I'll show you a diagram, but it's the one with the 3 fatty acid tails. And we divide them into fats and oils. Fats are the things that we get from animals. Oils typically are what we get from plants. And I'll show you the difference. It's a glycerol molecule with 3 fatty acid tails. The fatty acids can be saturated or unsaturated. For this class, we're not going to worry about it too much, but since I brought it up, I'll show you a diagram so you can see the difference. It turns out this is not nearly as important as we thought it was when it comes to health. The second category are the phospholipids. We talked about these when we were talking about cell membranes and the phospholipid bilayer. So it's the structure of the cell membrane. This is another term for cell membrane, is plasma membrane. It means the same thing. And then the third category are the steroids. When we say steroid, often people just think of the hormone, the steroid hormones, but cholesterol is a sterol or a steroid. And I'll show you what they all have in common. And testosterone and estrogen are both made off of a basic cholesterol skeleton. All right, so let's do the triglycerides first. The glycerol part is the 3-carbon unit over on the side. Here is the 3-carbon unit. Each one of the carbons here can hold on to one of those fatty acid tails. Here it's kind of abbreviated just with the, no, it doesn't matter. 3 fatty acid tails. This is why it's called a triglyceride. Tri for the 3 fatty acid tails and glycera for the glycerol over here. This is another representation of exactly the same thing. You can put the fatty acid tails. Notice the fatty acid tails are largely carbons and hydrogens. This is what makes this thing non-polar. Carbons and hydrogens share their electrons perfectly. There's no charge. Remember, we talked about polar and how some things are polar and non-polar. Okay, these bonds are non-polar. There's no difference. They share the electrons perfectly, so there's no partial electrical charges on anything. Whereas the other molecules, the carbohydrates, the proteins, and the nucleic acids, all of them have oxygen in there, and oxygen, if it's bound to anything other than another oxygen, it's a real electron hog, and it's not sharing the electrons. And that's where you end up with these polar bonds where there's a partial negative on one side and a partial positive on the other. This is not like that. These carbons and hydrogens share perfectly, completely neutral, electrically neutral. And so if you try to mix this with water, you can shake that up. If you've ever made salad dressing and you've got vinegar, which is largely water, and some oil, and you shake it up and you can shake it, and you shake it all day long, that is not going to get together. It very quickly will separate because polar and non-polar are not attracted to each other. All right, so more about fatty acids in detail. So here we can talk about the difference between saturated and unsaturated. In the saturated fatty acids, the carbons are all just holding hands, linked. They're linked to as many places as they can be. How to say this? Each carbon is making 4 bonds with 4 other atoms. These tend to be solid at room temperature. And then the unsaturated, that means that the carbons, instead of just having, holding hands individually with the carbons on either side of them. At some point, two of their bonds are coming in one direction, and so it puts a kink in the chain, as I will show you. So if there's a double bond, doesn't matter. These tend to be liquid at room temperature. This is what we refer to as oils. So let me show you. Here you've got a bunch of carbons. They're represented by the black balls, and then the white balls represent the hydrogens, and everything's all nicely lined up. These are butter, lard, for example. And then if you put a double bond here, this fatty acid is no longer a straight line. It's now kinked. These are liquid at room temperature. If you need a way of thinking about this, there's probably a place in your house somewhere where you have your books all piled on top of each other. And if the books are straight, these things will line up very easily. But if you try to throw in a 3-ring binder and the thing's kind of crooked, and you try to put your books on top of that, they're going to like slide off of each other. That's kind of what's happening here with these unsaturated fatty acids. They don't line up very easily with each other. If you mess around with the temperature, then all bets are off, because you can put oil in the fridge and it gets a little bit more solid, or you can heat up butter and it'll melt. But at room temperature, saturated fats tend to be solid, and oils or unsaturated oils tend to be liquid. And here's this representation. So carbons single bonded to each other, straight line. If you throw in a double bond here, you've got a kink in the chain. In case it ever comes up in your life, the way the carbons are numbered, the one that's attached first, remember the glycerols on one side, the one that's attached there is the alpha carbon, and the one at the other end is called the omega carbon. And so if you've ever heard of omega-3 fatty acids, it's talking of 3 bonds in, that's where the double bond is. Or an omega-6 fatty acid, that's where the double bond is. And phospholipids. We've talked about phospholipids before. Here are 3 different representations of phospholipids. There's a polar head. That means that there's a phosphate group, there's some oxygens, they're not sharing the electrons equally, so they're polar. Remember, there's a little bit of a positive charge, a little bit of a negative charge. This is going to interact beautifully with water. And then here you've got those carbons and hydrogens sharing the electrons perfectly, so this is non-polar. This stuff is not going to dissolve easily with water. And that's why the lipid tails face in towards each other, and then the phosphate heads face into the cell and out of the cell in the aqueous solution. And this is the super simplified version of it. There's the phosphate head, and then here are the lipid tails. And then finally, our last category, the steroids. They're lipids like triglycerides and phospholipids, in that they're largely made of carbons and hydrogens, which share their electrons well, so they're non-polar. They're different from the other two because there are these rings, and that's pretty much what they are. It's just this 4-ring structure. That's what they all have in common. So here's the basic cholesterol. And then this is what estrogen and testosterone are built off of. And what varies is the functional groups off of this. And these, having the different hormones, this different steroid hormones, have these enormous consequences for what structures get built up more than others. All right, so that was a quick and dirty review of all of the 4 macromolecules. We're going to put it into a table. Macromolecule name over on the left, the individual units of the macromolecule. This polymer column is not that important for our class, but then whether it's soluble in water or not. So the macromolecules that we saw were carbs, proteins, nucleic acids, and lipids. Lipids are the weird one. Lipids are the ones that have multiple categories within them. All right, so let's start with the carbohydrates. Individual monomers are monosaccharides. That means one sugar. Polymer, not hugely surprising, polysaccharide. This is simple sugars, you'll hear it called, versus complex carbohydrates. In both cases, they break down to, largely they break down to just a bunch of glucoses. Soluble in water, you bet. Proteins, the individual subunits, the monomers, are called amino acids. And if you put a bunch of them together, polypeptide or peptides. Soluble in water, you bet. Nucleic acids, the monomers are nucleotides. The polymer, I've never heard anybody use this, polynucleotide. And soluble in water, yeah. And then the lipids don't quite follow this pattern. So this monomer thing doesn't really apply, not applicable exactly. However, in our textbook for this class, it'll refer to the monomers as fatty acids. And you can kind of make that case because it's triglycerides, which is largely the lipids that we take in when we eat things. In digestion, we cut, we break the molecule apart. And remember, triglycerides have 3 of these. When I say not polymer, because polymers typically we're talking about hundreds or thousands of these monomers, and that's not the case with the lipids. At most, you've got 3 of these fatty acid tails. With the phospholipids, you've got 2. And the steroids, it doesn't really apply. All right, so the polymer, there's not really a big old polymer. And then the final question, soluble in water, no. And that's what puts all of these in the same category. They're not soluble in water. And voila, that is the summary of the big 4 macromolecules. And in our class, we'll talk primarily about carbs, proteins, and lipids, and how we digest those, how we break those down. I'll mention in passing nucleic acids, but like I said, mostly people make a big deal about carbs, proteins, and lipids. And I did want to make one more point, and that is these molecules largely are being used for one of 2 purposes. Either we need them for the fuel that they provide, the energy that they can provide us, or we use them for structure, for building ourselves. That expression, you are what you eat. It is literally true. You build your muscles, their bones, you build it out of the stuff that we eat. And I'll get back to that point during the regular lectures. All right, see you over there. This is the end of this part.